So it's my great privilege and honour to introduce Professor Peter Doherty, one of the world's premier immunovirologists. Peter is a vet by training, a Queenslander by birth, and gained his PhD in Scotland. While working at ANU with Rolf Zinkernagel, he made critical observations that allowed him to explain to us how foreign proteins are recognised by the T cell arm of the immune system. This so-called MHC restriction still underpins our understanding of defence against viruses, including HIV, hepatitis C, HPV, and guides the development of more effective vaccines to this day. He's gone on to dissect T cell-mediated immune response, particularly to flu, in a quest to improve vaccine efficacy and production. The quality and impact of his work has been recognised by multiple prizes, awards and fellowships, which include the Lasker Prize in 1995, the Nobel Prize for Physiology in 1996, and an Australian of the Year in 1997. And I think the latter is highly appropriate because Peter is not only defined by his intellect, but also by his dry wit and his self-deprecating humour, both archetypal characteristics of Australians of his era that we as a nation should value and emulate. I've been lucky enough to sit in a number of small meetings with Peter over the years and witness his ability to cut through, asking critical questions and proposing original, alternative approaches to difficult questions. In addition, his veterinary training has left him with an acute bullshit detector that keeps people on track. He has the great ability to engage with junior colleagues and has successfully mentored many. He has great communication skills and latterly has taken up as an author of several books, as he has developed a passion in communicating science to a wider audience. With this in mind, I truly look forward to his presentation entitled Living with Killers. Thank you, Peter.
Well, thanks very much, Tony. I had the privilege of working with Tony and with David Cooper some years back on a big program grant we had between Melbourne and Sydney and Canberra. And it was a great experience, and I developed an enormous respect for the group here and a great affection for them too, because this is a terrific human activity as well as a great science activity. And we've heard about that, but I couldn't be more supportive of that view. With respect to the deprecating personality, growing up in Queensland when I did, every sentence was kind of ended with, it was before Queenslanders started to raise their voice at the end of every sentence because of the uncertainty of the situation there. But every sentence ended with the phrase, a brief pause and then, but you can't win, mate. And as regards public policy and so forth, I'm definitely in that situation because I keep tweeting about the Abbott government. And this is our new building in Melbourne. That's actually the artist's impression, but there's actually a building there now. It's on the corner of Grattan Street and Royal Parade. It's a new infectious disease building, and it's a very unusual concept. We've got 3 floors of university microbiology and immunology department, which is the academic department, a very fine department. But we've also brought into that building the state virus diagnostic labs at Vidral, state bacteriology diagnostic labs, the infectious disease physicians from the hospitals who have lab work going on, tuberculosis centre, WHO influenza centre, university, state government responsibilities. It's trying to mix a lot of different cultures in the aim of bringing the science back to actual real disease problems and what's out there in the community. We've just achieved the recruitment of Sharon Lewin as director, and of course Sharon is running this International AIDS meeting, and she's held off from taking up the directorships till that's over. But with those different reporting responsibilities to the university, the state health department, part of Sharon's job will be herding cats. And if anyone has worked with Sharon at all, you'll know that nobody is better at that in the whole world. And it's wonderful to have her there. Now, my talk is not possibly all as scientific as some of the ones you've heard, because I know that Michael Kirby's here. And but I'll do my best. So we're constantly challenged by all sorts of infections that come out of nature. There are very large numbers of them, and of course we first became aware of HIV back in 1981, but it had been around for much longer than that, as we all realise. That's caused a pandemic, and that pandemic is still going on. It's a continuing pandemic, and I think it's going to continue for a very long time. But there are many other viruses that threaten us. One that we worry about at the moment is Chikungunya Virus, an insect-borne virus that's very active in the Indian Ocean, and it's now got across somehow into the Americas, and they're getting cases in the United States. And how these viruses move around is very intriguing, because where do they come from? Are they travelling in people's blood, or are the mosquitoes travelling on aeroplanes, or they're going on package vacations, or what is it? We don't really know. There are horrible pathogens out there. We all know about Hendra Virus. The virus was worked out principally at the Australian Animal Health Lab in Geelong. And you may have seen this movie, Contagion. It was described as the thinking person's horror movie. And if you want to see a hideous pandemic portrayed in a movie, rent it from the video store. Didn't do very well. It didn't have a really bad guy in it. You know, if you want to have an infectious disease horror movie, you need some really mad general or something that wants to do something totally nutty. But this is just a bad pathogen, and that's not nearly as appealing. We much more prefer to think about bad guys than bad bugs. So we've recently become aware that there are a whole spectrum of viruses out there in the bat population, fruit bats particularly. We didn't realise this before the SARS virus came along in 2001 or whenever it was. We didn't realise before Hendra Virus came along. We now know the Ebola virus, which is currently causing a terrible epidemic in Africa. Almost 1,000 people infected with Ebola. We've never seen that happen before. We didn't know that that's a disease of fruit bat. We're worried about a very similar virus, not coming out of fruit bats, but it seems out of camels in the Middle East. Or maybe it goes from bats to camels in the Middle East, the MERS virus, and we are worried that that could cause some sort of a global outbreak, because like the SARS virus, it spreads by respiratory infection, and it will spread from person to person. So there are constantly these challenges coming. And I'm talking about influenza, which is an incredibly infectious virus. Of course, if HIV was as infectious by the respiratory route as flu, we'd all be in really very serious problems. Fortunately, viruses do not normally change their mode of pathogenesis very much. HIV is, as you know, blood-borne pathogen. Flu isn't even, it's not only not a blood-borne infection, it's not even a systemic infection. It's a localized infection in the respiratory tract. Still, nonetheless, it will kill us very quickly, and it's a very bad disease. The other problem with flu is, and one of the reasons it's so incredibly infectious, is that when we're still feeling quite well, we've got an enormous amount of virus in when we cough or when we breathe. And so we tend to move around at a stage when we're infected with the virus, we're feeling okay, and flu really flies. It flies from city to city in aeroplanes, and that's the way the virus gets around. The 2009 Swine Flu Pandemic, which started probably in Mexico when two pig viruses got together and mixed up in a pig, as flu viruses do, and then and emerged in a way that was very infectious to humans. That virus was probably already in Australia before we even detected it in Mexico and California. So these things now go around the world with enormous speed, much, much faster than HIV. HIV is this grumbling pandemic which goes on, and the virus survives by persisting in us. Flu survives out there in nature because it infects multiple species, because it mutates all the time and all the rest of it. And it's a different type of challenge. It still retains the record for the biggest pandemic of the 20th century, the 50 million or so that died in the 1918-19, but of course it will be overtaken by HIV as that goes on. We've never had another flu pandemic as bad as that. Whether we would or not, we don't know, because therapy has improved so much, and many people who died in 1918-19 probably died from secondary bacterial infections. Of course, we can handle the bacterial side, and our supportive therapy would be much much better than it was then. But we still are concerned about it, and we still constantly raise the issue of influenza, especially if we need funding for influenza research. But it is a dangerous pathogen. The similarities and differences between influenza and HIV. They're both RNA viruses with poor proliferating mechanisms, or no proliferating mechanisms, so they're throwing off variants all the time. Same through of Hep C, of course. And because of that, if you're having a vaccination strategy, you've got to constantly change the character of your vaccine. The influenza viruses, as I said, are extremely infectious, and they're much more infectious than HIV. And they're localized to the respiratory tract, and the virus is generally cleared within 7 to 10 days, 12 days or so, maybe a bit longer depending on age of people and all the rest of it. And that means we can make effective vaccines against influenza, and we've had them for many years. A flu pandemic will go for 1 to 3 years. It then sort of burns out, the viruses mutate, we get variants of that virus that are circulating. We call these seasonal epidemics for some reason, even though the viruses are changed. The pandemic viruses come in from wildlife or from domestic animal species, and that really is the strategy that flu has for survival in nature. Influenza is basically a disease of aquatic birds. The virus survives very well in water. There's an enormous variety of these things in ducks and swans and geese and so forth. You can take almost any bird population throughout the world, or aquatic bird population, and find influenza viruses. Most of them are not dangerous. They're not particularly pathogenic in the birds themselves. But when they jump, and when they jump species, and when they mutate, and when they change, or they get together with a bad virus, then we can get an extremely bad pathogen. And it will come out of nature, and we've got no way of knowing what's coming out of or where it's coming from. And of course, HIV, again, as I said, is quite different in its pathogenesis. Now, the way that we handle infection is, of course, through our immune response. The word immune comes from the Latin immunus, which means without tax. This is where the Abbott government's going, by the way. No taxes, no regulation, no support for anything. But without tax, there was a category of Roman soldier called, they were in the genio immunium. And these were soldiers who'd returned from the wars, and for a time they paid no tax. And so that's where we get immunity. And it's various types of immunity. We've heard some of them mentioned in the talks. We heard particularly Francoise earlier on talk about natural killers and so forth. What we're talking about there is innate immunity, the early acute response that's brought on and produces all sorts of, there are various types of cells involved in this that will deliver molecules like gamma interferon. There are various types of what we call cytokines, chemokines, toxic molecules, which will help to hold an infection in check in that earlier stage. There's been quite a lot of work on this with HIV over the last few years. I was chair of the board of the CHAVI, the big group in that's run from Duke University in California. And there's a lot of stuff going on there in this sort of area. It's extremely important. It's an extremely important component of the immune system. It will never stop the infection in its tracks, but it keeps it under control, often in that early stage. And probably in its early stages, HIV is not that different from any other infection, really. It gets in, turns on the immune response, turns on these types of responses, and goes through that early phase. This is a very old system in evolutionary terms. The natural killer cells and some of the genes that we associate with that right through evolution. The toll-like receptors, for instance, we associate with this were first discovered in fruit flies. So this is a very old biological, these are very old biological systems. Adaptive immunity, which is the part of the immune response that we're priming up with the vaccination, is about 350 million years old. We first see it in the jawed fishes, though there are elements of it in the lampreys, which are a little bit more primitive. And this is the part of immunity that we use a vaccine to generate immunological memory. We generate immunological memories for Cd8, Cd4 T cells, and certainly for the antibody-forming cells, the B cells. And that is the basis of vaccination. We have vaccines against influenza. We can vaccinate against influenza. It's a reasonably good vaccine. It's not great, not particularly good in the elderly, but then, you know, there are too many old people and the world is overpopulated. And Michael, we have to make some sacrifices. And vaccines, the two viruses that have been eliminated from the planet are smallpox and rinderpest, a cattle disease called rinderpest. Both have been eliminated by very straightforward and rather primitive almost vaccines. Nothing very high tech. It was just giving them intelligently and running a program intelligently. Now, rinderpest is the direct cousin of measles virus. They diverged about 1,000 years back, or, you know, we didn't have a lot of virology going on then, but that's what the sort of archaeology says. About 1,000 years ago, they separated, and it could be that we got measles from cattle when we started to come into communities and agricultural communities and all the rest of it. Now, we could eliminate measles. Why haven't we eliminated measles? Because people won't vaccinate their kids against measles. We keep getting regular outbreaks, and it's not a third world problem, it's a first world problem, because people in the first world will not vaccinate their children. Now, it's all to do with the rise of empowerment and individualism and the internet and all the rest of it, but it's a reality. We can't get rid of poliovirus. Again, a virus which could be perfectly readily eliminated, but we can't get rid of it because vaccine workers have been killed in Nigeria and in Pakistan, and because, in actual fact, they've gone back to using live vaccine, which the H7n9 vaccine, where you can actually get virulent flu virus back in the stools of people giving the vaccine. And this has been picked up in Israel. Vince Racaniello was here, I think, about 2 weeks ago, talking about that at the ASM meeting. So I think you're going to be very lucky to get rid of HIV or Hep C globally because of the differences in perceptions, the social context, and all the rest of it. So the best we can do is get things as widespread as possible. And getting a vaccine for HIV, HCV, or HIV is, to my mind, almost impossible, and we need some major conceptual breakthrough. The one that we've all been pinning our hopes on, both for getting a cross-reactive influenza vaccine and an HIV vaccine, are the cross-reactive antibodies that we can find if we look very hard. About 1 in a million plasma cells is producing cross-reactive antibodies, which would neutralize across a whole spectrum of HIV or of influenza. We can find exactly the same thing. But the issue is that these are not primary antibodies generated first off in an immune response. They arise by a random stochastic process of somatic mutation. And we don't know how to make an immune system make those antibodies. If we could work that out, maybe we would have some good chance of getting something reasonable in the sense of an HIV vaccine. And of course, vaccines are the cheapest way to deal with an infection. It's one of the great triumphs of medicine, vaccination. A lot of it was by, I guess, Louis Pasteur really started a lot of it off. But a vaccine would be great, but I don't see it happening anytime soon. What I work on is the killer T cell, as was mentioned. If you're thinking of the Roman legionaries, these are actually the legionnaires, because these are the guys, the legionnaire carried a short stabbing sword, and they had a big shield. And when they were being attacked by a lot of Scotsmen, for instance, who had swords about 4 feet long and were waving them above their head, they just put these shields up, and when the Scotsman got too close, they poked him in the tummy. And that's more or less the way that the cytotoxic or killer or Cd8 T cell that I work on actually operates. The dog in armour is just a bad immunological joke, and I won't tell you about it. Science jokes are terrible. This is a killer T cell. Watch it quickly. That's a normal cell. The green guy's the killer, and it's going to kill that. As it turns pink, its membrane is being destroyed, and you can see that cell is killed, right? No doubt about that. That's a pretty dead cell. They're actually not killer T cells. They're inducers of suicide cells because they turn the cell on to commit suicide. I've just realized they're also necrophiliac because they stick around. These were done by Misty. Misty, it was though she looks rather blonde, and she was attacked by Andrew Bolt. She's one of the people attacked by Andrew Bolt. She's actually a Curie scholar, did wonderfully well in Cambridge, and is now back at the Peter Mac in Melbourne. Now, With the antibodies that are produced by the plasma cells and secreted into the blood, they bind to the virus itself. They'll bind to influenza virus surface proteins or HIV surface proteins, and it's a protein-protein interaction. The killer T cell, or the T cell, is focused onto the cell. So if you think the virus is this big, the cell is this big, and it's a factory that's producing those virus particles. So what the killer T cell does is it knocks off the factory. It's not the fighter plane, it's the bomber plane, if you like. It gets rid of the factory that's producing virus particles. And the way that it's focused onto the cell, and you would have heard Francois mention HLA, which is a transplantation molecule, is it focuses on to a bit of the virus, a virus peptide that is expressed on the surface of the cell, bound into the tip of the transplantation molecule. This is from some of our recent work where we did co-crystallization of T cell receptors and the MHC molecule. So here's your peptide, about 8 or 9 amino acids, exactly the same in HIV. Cd8 T cells turn on early in HIV. As Francois told us, if you've got a very good Cd8 T cell response, you will control that pretty well for a time. But of course, you can't control it indefinitely in most cases. But here's the peptide, a little bit of the virus that's split up and then it's carried to cell surface. It's called the transplantation system because it was discovered by a transplantation. It was discovered when people did grafting of tissues and tried to transplant kidneys and all the rest of it. In fact, it's actually a self-surveillance system which manifests as a problem in transplantation. Now, a lot of the peptides from the virus that are seen in influenza and in HIV and in many infections actually come from internal proteins of the virus, not the ones on the outside that are recognized by the antibodies and are very susceptible to antibody-mediated selection and change, but internal proteins. They do change, and that's evidence that they're actually being selected, of course, because mutants are being selected. But because of that, with influenza, we get infected, as you know, with different types of influenza. And we get infected with variants of the same influenza as the surface molecules change, the seasonal epidemics. In each of those events, we're often priming up a lot of cross-reactive T cells that will recognize a new influenza variant that comes along if we've still got those cross-reactive peptides in that flu virus. And we often do. There's a lot of conservation of these flu peptides across the influenza viruses. That means that people who have good Cd8 T cell immunity to influenza are often somewhat protected against novel flu variants. And with flu, it's not like HIV where you've got to have something that goes on indefinitely. With flu, all you need is that protection working over the first 5 or 7 days. And if you've got enhanced protection there, and you can actually eliminate the virus a bit more quickly, you can do much better with flu than you would otherwise. And that really is part of the challenge with flu. So with flu, we do have the possibility of using a cross-reactive T cell vaccine as a prophylactic vaccine. It wouldn't stop you getting infected. It might not stop you getting sick even, but it might stop you getting dead, which is good. And so, but with HIV, when people became aware of this and they thought about it, they tried very hard to make Cd8 CTL HIV vaccines. A lot of effort went into it. There was some early prominence, particularly from monkey studies done by Norm Lettvin and others, but in the end analysis, it just doesn't work. It doesn't mean that it might not be helpful to do that if you could also make that antibody response that I talked about. It would be good to have as many mechanisms coming into the system as possible, because with all these viruses, even though you can isolate different types of effective mechanisms, if you've got them all working together, it's better. I mean, it always works better. Particularly true of the big viruses, like the herpes viruses. You need CD4 T cells, CD8 antibodies, all the rest of it. So there is some hope for a strategy there. Now, of course, any immune response involves clonal expansion for a very small number of precursor cells that have got the right receptors for that peptide MHC. If you're talking about CD8 T cells, you get clonal expansion, differentiation, and then some of those go on to give you immunological memory. And immunological memory is what we're looking for in all vaccination. This is Cd8 T cell immunological memory for influenza. These experiments were done initially in mice. We've been doing experiments like that in humans, looking at cytomegalovirus-specific T cells. We're all persistently infected with this thing, and as people go through life, you can actually trace the particular lineage of cells, because you can identify the T cell receptors looking at using a single cell PCR type approach, and you can actually trace the clonotypes through in the very, very long term. And there's a lot of scope for doing that more in influenza. Some of that work's been done at, say the vaccine centre in NIH with HIV. But we've worked out how to actually identify the two chains of the T cell receptor in single cells taken directly from a mouse or taken directly from human arm, and then trace those clones through over a number of times if you want to study memory. So it's a really good technology for doing that. Now, with respect to these Cd8 T cell responses, I'll just mention this. What we've found in influenza studies, or what Catherine Kazierska has found in some experiments a couple of years back, is that as we age, we lose our capacity to respond to new things. If you're a Liberal Party voter, you lose your capacity to respond to new ideas very early, but sorry, I shouldn't be so politically partisan. But you lose your capacity to respond to new things in the immune sense. And we found that with the Cd8 T cells at least, you can stop that loss by priming very early. So what we actually need to do, I think, with a lot of these infections, where we think we want to have a Cd8 T cell component, is avoid that loss equation by doing whatever priming you want to do very early on. Now, unfortunately, influenza virus vaccines, antibody-based vaccines, they stop the actual natural priming process if they work, because they stop the infection. And it's infection that primes, say, the Cd8 T cell side of things. But priming early is probably important if we're thinking about any sort of vaccination strategy that involves Cd8 T cells whatsoever. Here's some more. These are memory T cells in the skin. Some of these memory T cells sit around in local sites. They're called resting memory. But if that's resting, it's certainly not a couch potato type status. I mean, you can see that they're moving around all the time. And of course, the whole body is very dynamic. This is interesting because it's a herpes virus T cell immunity. And Scott Mueller, who did the study, is actually taking the photographs. He's photographing these cells in the skin. You can use these two photo techniques to actually photograph cells in the body and see them moving. This has given us a much better understanding of how T cells actually operate in lymph nodes and so forth, and it's really been a very exciting story, pioneered by people like Ron Germain at the NIH. Now, with flu, you infect intranasally, the virus gets eliminated. If the mice are primed, these are virus-specific killer T cells coming into the lung. And it's these we're looking at. In a primary infection, they'll come in after about 7 days. This is starting right from the beginning, where we've got very few precursor cells. After about 7 days, we're getting the T cells into the lung, and the virus is eliminated between here and here, between day 7 and 10. And if you take out the other components of the immune system, the CD4 T cells and the antibodies, the Cd8 T cells will clear the virus. If you take out the Cd8 T cells, the CD4 T cells and the antibodies will clear the virus. So this is not the only mechanism. But you can see here, these mice were primed up with a cross-reactive virus about 7 months back, which is about a quarter of the lifespan of a lab mouse. And you can see that there's a much bigger response, but it's not there as early as day 3. And that's the problem with Cd8 T cell responses. You have to turn them back on, and that's why they didn't work with HIV. Because even if no matter how good you get these things, there's still a window where the virus can get in and get established before these T cells can turn on, do their job, and start to control the infection. You can see how much bigger that secondary response is than the primary response. And this is the virus being eliminated in the primary response, and that's the virus being eliminated in in the secondary response. Now that protects the mouse. Or if you boost it up even further, you can really stop them getting sick at all, even if you give a very, very highly lethal virus. But the virus will still grow just as much over the first 24 hours. And that's the issue of trying to use this as a sterilizing vaccine. Is there evidence that this actually works in humans? Yes, there is. There's various evidence over the years. It started with Andrew McMichael way back, who was doing studies on this, and then HIV came along. And what happened with a lot of the good virologists, immunologists who were working with humans is they all went off to study HIV because it was such a crisis and it was happening so quickly. And so there was not a lot done for a long time. And then influenza came back. And under the George W. Bush administration, we got very frightened about the H5N1 bird flu. And even members of the Republican Party realized that rich people could be killed by flu. And so we had to do something, and we had to do it fast. And so a lot of money went into flu. That's a bit unfair to Bush, because I think, you know, one of the great legacies, or perhaps the great legacy of George W. Bush, who doesn't always get positive remarks, is really PEPFAR. And he did that, and he's enormously well regarded in countries where that has been such a help. So, you know, we never quite know who our friends and enemies are. And for the Australians, of course, one would mention the Palmer United Party. But, to finish up the story, because I don't, I'm already later than I should be. I was told 1 o'clock for lunch. Yeah, okay, I'll be absolutely finished. Again, Catherine has gone across completely into human studies of influenza in humans, and we're shifting a lot of the lab into human work because you can now do so much great science in humans with modern molecular technology, because you can work with very, very small numbers of cells. You can't do experiments, obviously, but you can do all sorts of analyses that are really terrific. And actually, if you want to look at immunological memory, it's really humans are the best system to do that. She got involved with the group in Shanghai that was looking at the H7n9 virus. Now, H7n9 is another one of those flu viruses that comes from birds, gets into humans, and is highly lethal. And it was infecting and killing about 33% of the people it was infecting. Mainly men, and particularly susceptible were older men. Partly because they thought that the retired men were being sent out to do the shopping, and part of the shopping is to go and buy live birds from the bird market and then bring them home and kill them. And that's probably why they were contracting the infection. And so it's really a bad infection. But the group in Shanghai, a very good clinical group, took a lot of samples, and they looked at these very closely. And then Catherine did a lot of the cellular immunology on it, and we now have a very nice ongoing collaboration with them. And of course, that's part of what's happening with Australia. We're looking much more to our north, getting a lot more collaboration going on in all sorts of situations. We've heard some of it with HIV, we're expanding it with influenza, and it's a really great thing, and I think that's one of the best things we can do. Here's our serial, you can see them as serial killers. And again, we're looking at the killer T cells. You see, they'll kill and kill and kill, so we need to finish. Well, what she found is that in these H7n9 viruses, which is a virus we've never seen before, and we've never seen it in humans, but I don't think we've even seen it in chickens, but they have a lot of the conserved peptides, the ones that are present in most of the other flu viruses. And that means that being in China, of course, people are constantly infected with flu, as everywhere, and a lot of these people will be primed up against those conserved peptides. So they'll have a primed Cd8 response. And this is the way it worked out. It was found that anyone who had what's called A2, HLA-A2, which is a common allele in Caucasians, would have these cross-reactive T cells. On the other hand, if you have the HLA-A24, which is common in some Alaskan groups and is common in our indigenous population, we didn't find any of these cross-reactive peptides that would bind to A24. And the Alaskans, if you look at 1918-19, for instance, the Australian indigenous population and the Alaskans were very, very susceptible to influenza. There were a lot of deaths. In fact, one of the resurrection of that virus used someone who died quickly in Alaska. And if you look at the different groups, for the ones who've got this cross-reactive immunity, here's Caucasoid and so forth, and you can see The Australian Indigenous and the Alaskan Yupik are right down at this extreme. So we haven't really looked within those populations yet. Catherine has a substantial grant working with people in Darwin with all the usual involvement from the communities and so forth to look at this in our Australian Indigenous populations. But this is what the actual mapping would show us, that the one reason why they may be much more susceptible is because of this HLA allele. And this is looking at the white blood cells that they'd taken from these patients and looking at them shortly after they were hospitalised and then through the infection. And you can see the ones that recovered made very big Cd8 T cell responses very early, not much CD4 T cell response. The ones that recovered more slowly, Cd8 T cells and CD4 T cells. And that sort of says to me is that Cd8 T cells weren't handling it quickly, more stimulation was going on, so we brought the CD4 T cell numbers up. This is a bit later, and again, people that recovered. And the ones that died had lousy responses. They really didn't do much at all. So it does point to the fact that these Cd8 T cell responses are indeed protective, and that's where I'll finish. It can be protective. They can eliminate flu, and they do control HIV for a substantial time. And of course, in those high responders, you get very good control for quite a while. And it's likely that CTL vaccines, certainly in flu, could decrease mortality significantly. And whether there's an economic case for doing it, of course, is another matter. But, they may help with flu if we can get something better on the antibody side. So thanks very much.